Thank you for inviting me, and thank you for participating in this talk. As I have seen the program of this winter school, it's focusing mostly on high-level quantum chemical or quantum mechanical calculations. And this talk will be unique in this series, and it will introduce a novel area, at least as I have seen the previous year's program. Drug discovery was not touched by this winter school so far, but I hope that in the future there will be more and more talks that are focusing on the application of these theoretical studies. Today I prepared with a presentation that focuses on the computational tools for drug discovery. I want to introduce you the field itself, the challenges and the possible solutions. And I apologize that this will not be a theoretical presentation. In most of the cases, I will focus on the results, the outcomes, and the whys, why we are doing these type of approaches to achieve the goals of drug discovery. So it will be a bird eye view of the tools that are applied in the industry. And I'm from ChemAxon. ChemAxon is a cheminformatics tool developer company that's a gold standard in the industry for storing and retrieving chemical information and providing chemical intelligence to various environments. And in this talk, I will talk about the topic in general and not focusing only on the tools that ChemAxon provides. So let's start with the motivation. Pharmaceutical research is focusing on delivering therapies, solutions to contribute to human welfare in general. But the drug itself should be safe and it should have efficacy, high efficacy on the targets to be able to cure the disease. Safety is related to the adverse effects. It should be minimized as much as possible, and the efficacy should be high so that it can interact with the target protein or the system itself to express its action on the system. And these two, the safety and efficacy, are the main motivations where we want to design novel compounds and balance the attributes of the compounds. What I show here is the pharmacokinetic curve of a drug to introduce you the system we are working on. When a xenobiotic or a drug is administered, then the concentration will increase in blood, then it will decrease, and you can also calculate the area under the curve. And it should reach the concentration, which is the minimal effective concentration, where we can achieve target engagement and where it can modulate the pathological process to cure the disease. And there is another concentration that is correlated with the safety part, the minimal tolerable concentration, where it will start interacting with various other proteins, membranes, biological systems that will have the adverse effects. So in between these two, there is this therapeutic range or therapeutic window that we should optimize for to be as high as possible. So the pharmacokinetics is about this absorption, distribution, metabolism of the drug that's administered, and pharmacodynamics is related to the effect, which is primarily the concentration of the drug in the milieu where it needs to interact with the protein. And you can combine these two to have the pharmacokinetic, pharmacodynamic profile and the curved course of the changes in time. So that's this is the main point. And the human body is complex. It has high complexity. When a drug is taken orally, then it will be in the system where it needs to dissolve to be able to be in the fluids environment. Then it passes the gut wall to get into the portal vein, where it will go first to the liver. There will happen the metabolism, the first part, the metabolism of the drug, and then it can reach its therapeutic target. So the process itself involves multiple proteins, membranes, physical chemical processes. And if the drug cannot reach its target and it cannot bind to the targets, then it will not have efficacy. So if in the test tube it reacts with high efficacy, with high affinity to the target, but if in the body it cannot reach its target, then you will observe low efficacy. And this leads to the topic of anti-targets and the problem of optimizing the absorption, distribution, metabolism, and excretion, which is abbreviated as ADME in drug discovery. And this is an optimization problem against the biological system, the human body, where if you have pure systematic exposure, then it might be related to the distribution, where blood-brain barrier penetration might be a problem, or the drugs, when it's in the bloodstream, it can bind to human serum albumin, and then its free concentration will decrease significantly. So human serum albumin binding can be an anti-target. If it is a substrate of transporters, there are multiple transporters in the human body that tries to prevent the body from xenobiotics. One of the examples is PGP, which is an ABC transporter, which is kind of a hydrophobic vacuum cleaner in the system that tries to prevent, especially the brain, by an efflux transport from the membrane, and it prevents penetrating to the human body. But if the clearance is high, that might be due to the metabolism that's mainly mediated by the cytochrome P450 enzyme family, where there are different isoforms available, that's shown here, and this can degrade the xenobiotic in the blood or in the body. Or there are factors that related to the induction, further synthesis of these enzymes that will degrade the drug. One of them is PXR or CAR. These are having hydrophobic packets that are less selective, so that they can bind the xenobiotics, and then they can scale up the production of metabolic enzymes that will degrade these xenobiotics in the body. If it is a pure bioavailability, it might be due still to the hepatic clearance or to the absorption. That might be due to the fact of pKa, solubility, lipophilicity. Log P is the octanol water partitioning coefficient, or the log of these partitioning coefficients, which is a key parameter in the drug discovery in general. In several in vivo studies, it was shown that log P correlates with toxicological outcomes. But the membrane penetration, how a molecule will go through a cell, is also an important factor. So we have several anti-targets in our system, while in principle, and in case of target-based drug discovery, there is another branch, the phenotypic screening. But in most of the cases, we know what the target is. And we have in most of the cases, only one target to engage with, while we have several others, up to approximately 20K proteins in the human body that we want to avoid the interaction with, to decrease the adverse effects and toxicological outcomes. So that's a challenge for drug discovery, to design molecules that are specific to a binding site. It avoids binding to the rest of the proteome and only targets one binding site with specific interactions. So this concludes to a problem of solving a Rubik cube. In case of medicinal chemistry optimizations, a compound is changed with chemical modifications. And when you make a chemical modifications, you change an atom, a hydrogen to a methyl, or you introduce a nitrogen to a heteroaromatic ring. This is an analogy of turning the Rubik cube that will trigger a change in the biological activity profile, and it will influence binding of this large number of targets and your protein target itself. So Rubik cubes has a vast amount of configurations that it can adopt. Up to 10 to the power of 19 configurations can be done with this simple cube, but it's in challenges, it's solved within 10 seconds by experts who are aware of the methods, how to turn the Rubik cube. And it's estimated that from all configurations in 20 steps, the cube can be solved. The druggable chemical space is a bit higher, but if you have proper tools that help you in the turns, then you will be able to get to the balanced safety and efficacy profile quicker, in a more cost-effective way. And therefore, and this is the demand for computational tools that can analyze the data, that can rely on physics-based methods to be able to help at every and each turn the medicinal chemists how to change the structure of interest. So our ultimate goal is a function that relies on predictive properties, and it's that relies on properties that can be calculated before the molecule is synthesized. That's the X. This is the descriptors that we can virtually calculate, or it can be the physics-based method itself, and it predicts the Y, the binding affinity. So how this is embedded into the drug discovery pipeline? Drug discovery starts with identifying the target itself, which is a biological activity, but it also includes computational tools when the whole proteome is screened, but that's out of the scope of today's talk. And then tests are developed where they can check in the biological system how the compound behaves. And there is a synthesis of the treatment, which is this turning the Rubik cube, making different modifications of the ligands to be more and more efficient to the target and have higher affinity. And then there are further testing in vitro that's for safety, to measure the profile that will be in the human in the ADME tox experiments, then testing in vivo, and then testing in man. In vivo is mostly animal tests. But the pipeline is enabled by computational tools in a way that there is a starting molecule with a hypothesis, and then you design alternatives, you synthesize the compound, test it with your assay cascades, and then there will be an analysis of the data that includes chemical intuition, team intelligence, QSAR, which is quantitative structure activity relationship, which is data mining and coming up with equations that can predict the target, and even including machine learning. So there is a learning, and then you will have a new hypothetical compound, a new hypothesis about the binding sites, the characteristics of the ligands that drives the profile itself. This is called, in most of the cases, DMTA cycle: design, make, test, and analyze cycle, where we have to solve the challenge. And the challenge is the protein-ligand interaction. This is a really simple example when we have a protein structure and it needs to interact with the inhibitor itself, and this will form a complex. So you can see the dissociation constant that will be the delta G, the free energy change, and that will correlate with the logarithm of this dissociation constant. But what happens under the hood when protein-ligand interactions are formed? We have a target of interest, and that's simplified with this green field here, which is in solution, so it's fully solvated by water molecules, and also our ligand is solvated with water molecules. Then it will form the interaction, it will bind to the binding site, to its pocket, and then the system will be rehydrated again. On this graph in the right hand side, you see the blue bar is the delta G change. And with ITC or several measurements, the enthalpy and the entropy contribution can be measured separately. And the enthalpy is driven by these non-bonding interactions, hydrogen bonds, van der Waals contacts, and polar dissolution in a simplified model, while the entropy part, the red, is the structuring, the change of the protein, flexibility, freedom to move, and the dissolution. So as you can see, the system is complex. We have to take into account the solvation plus the movements of the protein plus the interactions that are in the system. About the scale of these interactions, as I introduced in the pharma industry, IC50, EC50 effective concentration, inhibitory concentration, or Ki values are measured. And here I just created this simple chart to show you that what's the molar concentration with prefix, what's the delta G with kilojoule per mole or kcal per mole, and what's the affinity. So as you can see, 1 micromolar is a medium activity with 8.2 kcal per mole. Strong interactions are 12.3 kcal per mole. So there is a small change that will turn a ligand from medium binding to strong binding, and therefore we need methods that can predict within 1 kcal per mole the affinity to be able to use for prioritizing compounds. And on this example, I show you the intricate nature of this binding process. You see here this ligand with 2 methoxy groups, this 3-cycle, and here a phenyl ring. And on this phenyl ring, if the R is hydrogen, then the binding affinity, the Kd, is 200 nanomolar, which is somewhere here at about 9 kcal per mole. But if you change the hydrogen to methyl, a tiny change on this complex structure, the affinity will increase significantly, approximately 2 orders of magnitude. And if you change one of the hydrogens to fluorine, it will be 0.3 nanomolar. So from 200, we will go below 1 nanomolar. It's a tiny change. In the pockets, it will form multiple interactions. As you can see, this fluorine will be in proximity of hydrogens, and that will influence the binding significantly. Another example is this ligand here, which is even bigger. And this red part, this propyl part, protrudes or faces a small pocket that's surrounded by tyrosine, phenylalanine, and tryptophan, which is a lipophilic small cavity. And if it is a propyl, then the binding, the Ki will be 1 nanomolar. So it's 12.3 kcal per mole. But if you change this isopropyl group to hydrogen here, then the affinity will drop to 40 micromolar concentration, which is somewhere here. So as you can see, from roughly 6 kcal per mole, changing a hydrogen to 3 carbon atoms, you will get interaction of 12.3 kcal per mole. And this is due to the fact that this cavity, if it is a hydrogen, then there are no vacuum in the system, so there will be a water molecule inside this hole, which will be solvated by apolar groups, which is not feasible thermodynamically. and kicking out this water molecule with these 3 atoms will be beneficial, since this lipophilic region will be solvated in a lipophilic milieu, while the water molecule can go back to the bulk phase, where it will form hydrogen bonds with other water molecules. Another aspect of how much affinity can be gained by filling a cavity is shown on this example. Again, a large structure where the 2 R's can be hydrogen, fluorine, chlorine, or methyl. And for 3 of them, there are X-ray structures available, and this was docked into the binding pocket. And as you can see, if you increase the size of the R1, R2, then the binding affinity will increase significantly from 11.3 nanomolar to 0.14 nanomolar, which is 2.7 kcal per mole change, only due to the fact that instead of the hydrogen, fluorine, chlorine, or methyl will be perfectly matching the binding sites, and that will drive the interaction. So if you imagine the whole complex system with the protein, with the ligands, with all of these heteroatoms, all of the dissolution terms, tiny changes will have large influence. And in this case, it was also followed by ITC measurements, and I recommend to check this publication. The delta G seems to correlate with the van der Waals radii of the molecules quite nicely. But if you change the delta H, the enthalpy and entropy term, then you will see that it's under the hood, it's more complex, since it's not just the delta H will decrease nicely, but due to the structuring, due to the limited mobility of the ligand when it's more perfectly bound to the binding pocket, the first change will be entropically disfavored. And the last topic, as an introduction, is the non-additivity. If you have a ligand like this, and if you change at this part, the ligand 1 here, to ligand 2 by introducing a phenyl moiety, then the delta G, so the binding affinity change, will be 2.1 kcal per mole. And if you introduce a nitrogen, a basic nitrogen here, then the delta G will be 1.5 kcal per mole, which, if you do both changes, you can expect, or you can anticipate 3.3 kcal per mole change. But it was observed that it's 4.3. So it's not like playing with Lego elements in the optimization. Due to the entropy and enthalpic changes, not just the newly formed interactions have a role, but also the dissolution and also the structuring. And how about the molecules themselves, the ligands? Ligands are like chameleons. They are adapting due to the nature of chemistry. And again, sorry for that, this is a textbook example, but the environment, the pH, the solvent, the temperature impacts tautomerization, ionization, microspecies distribution, solubility, or lipophilicity. And if you think just about a simple amino acid like histidine, it has two basic, it has two strong, or the strongest pKs are, one of them is basic and the other is acidic. But there are two additional ionizable moieties in the system. So if you plot, and that's, or calculated properties, if you plot the microspecies distribution for this simple molecule, then you will see that there is a constant change in the microspecies that can bound to the protein target. At 7.4 pH, you will observe this green one, which is not having a net charge, with zero net charge. It's one of the amines positively charged, the carboxylic is negatively charged, but you will also have this other microspecies in the system where you have two positive and one negative charge. So when you are thinking of what to dock into a binding site, what to start evaluating, you have to think of all of these aspects, including tautomerization as well. where again, this is a simple calculation on this molecule, an oxoanol tautomerization, or two different ones can happen, and this will change the characteristics of the molecule, the number of hydrogen bonds it can form, the type of the hydrogen bonds it can form. This is an H bond donor. This is an H bond acceptor that will be significantly different in a binding site. And to show you an example, this is a recent publication from Syngenta. They revamped their tautomerization rule set last year. They had CBR14 as their primary rule set, how the molecules that are registered in Syngenta compound collection are to be represented. And then they carried out QM calculations using DFT calculations, B3LYP calculations with standard basis set for the pairs of the tautomers, even in vacuum, so without the solvation. And then they generated a new set of rules how the molecules should be represented in their compound collection, and this rule set is about 1,200 new rules. And when they checked the calculated properties with the old rule set, the CBR14, in some cases, like the pK calculation, there was absolutely no correlation in between the measured and the predicted pK. However, when they changed to this new rule set and applied the pK calculation on the novel tautomers that are correct or corrected, the R square increased significantly. And that was true for C log P calculation and 2 pK calculations itself. So only by applying some quantum chemical calculations, that's an approximation, you can get much higher accuracy when you're comparing another in silico to measured data. So I've shown you that there are a lot of challenges in the field, but we are lucky since the fundamental laws necessary for the mathematical treatment of a large part of physics and the whole chemistry are known. The difficulty lies only in the fact that application of these laws leads to equations that are too complex to be solved. And we know this almost for 100 years now. And I'm sure that all of you who are attending this conference have read this quote from Paul Dirac. So we have tools. One of the tool set is the physics-based approaches, where the level of approximations is about binding, or is about separating the system. There can be calculations that are ligand only, that are focusing on the ligands, electrostatic potential charge calculations or isosurfaces, Torsial scans, calculating tautomers or interactions, molecular mechanics for flexible, rigid alignment. So in the X-ray structure, if you have a complex with one of the ligands in the protein structure, what you observe, you can overlay your test compounds and check whether it fits in 3D. Can it adopt similar 3D conformation? Or you can apply flexible ligand and rigid protein models like docking, where you will apply scoring functions. Since I have seen that this conference is mainly on QM calculations, I try to squeeze in as much examples from the literature as possible to show applications of QM calculations. And I recommend to check this publication. It was in JMAT Chem a couple of years ago, where they investigated the role of halogen bonding, and they calculated various halogen derivatives of benzene, fluorobenzene, bromobenzene, and iodobenzene. And here what you see, this is the ESP isosurface representations calculated with MP2. And the red field corresponds to the positive charge. And as you can see, this is the effect of the sigma hole that's from bromo or chloro derivative. Here there is a positive region, a positive charge that can interact differently with proteins, and they calculated on surrogate systems the binding energy as well, and screened all the possible configurations with both with distance and with angle, with different angles, and calculated the interaction potential surface for this amide or backbone amide and methionine parts of the protein. And this led to fine-tuning the docking scoring functions with consideration of the halogen bonds themselves. Another application of QM methods, what I want to cite here is the site of metabolism prediction, the smart tip algorithm, where the group calculated QM energies for transitional states and from starting state, and from that the difference, the activation energy or barrier was calculated, again with small organic compounds, and the porphyrin ring that's activated, that's the compound one state of it, and from that derived these activation energies that are associated with these substructures of larger ligands. And from that, this is expressed in so-called SMART language, which is a substructure query, and derived rules. Also extended this with accessibility descriptor, where in the graph you will find that particular SMART pattern, and build a score out of it. Altogether, almost 140 calculations of the transitional states were done, and that resulted in 41 rules. And the smart tip algorithm could predict with the highest rank in 65% of the cases the metabolic side. So if you can remember, this is the ADME field, this is the efficacy field, whether the compound should be will be metabolized. And when it's metabolized, the scientist needs information, what's the site of metabolism, so that they can protect that position. And in this case, the readout is what's the metabolic position. And this tool could achieve 81% precision if you consider the top 3 ranked positions, relying on quantum chemical calculation extended with accessibility. With regards to the protein-ligand interactions, docking is the workhorse for virtual screening. Docking is the conformational search within the binding site of the protein. And this scheme shows you a quick overview of how the docking works. This is again oversimplification. You have an initial structure, you enumerate conformers, rotations of the conformers, and then transitions of the conformers, fill up a box with these translations, and then place it into the protein structure, avoid or discard the positions that are overlapping, that are clashing, and the ones that are not forming any interaction, and then score these possible conformations, and then optimize the final structure in a way that the protein is kept rigid, and you want to find the best binding pose. So this is the prototypical way how a grid-based search works for docking. And if you have the ligand and the receptor complex, you will have an array of possible solutions, and you need a scoring function. There are 4 major types of the scoring functions. There can be energy-based or physics-based that takes the van der Waals and the Coulomb interaction into account. There are empirical scoring functions, since what I said about the complexity, that takes into account the rotational bonds, the hydrophilic interactions, the van der Waals contacts, H-bond contacts, and this is fitted against a large set of observed protein-ligand complexes with known affinity data. So it has a bias, or it has intercept, and these interactions are also based on distance and angle in the dot pose and gives the score. There is another method called knowledge-based, when from the observed X-ray structures, atom pairs are gathered, and based on the distribution of these atom pairs, a PMF can be calculated. And there are machine learning methods that can take into account various descriptors to come up with the functions that will predict the energy of binding. Docking is not good enough in terms of energy prediction. Therefore, in most of the cases, it's used for virtual screening, where for the validation, there are two methods that's used. One is this accumulation curve. You rank the database, and if you find an active molecule, it's primarily a classification method. You have actives and non-actives when you are validating the model itself. When you find an active, you step one up and one right, and then if it is an inactive, only to the right. And that will show you the accumulation curve, and you can calculate the area under the curve. The other method, what I would prefer, is the ROC, the retrieval operational curve, when if you find an active, you step up. If you find an inactive, step to the right. So if you find all the actives, if you outrank the actives over the inactives, then the area under the ROC curve will be 1. The other method to validate the docking is called enrichment factor, and it has a Boltzmann enriched, the bedrock version as well, where you have a subset, you have a database that you want to rank. You prioritize the compounds based on the docking scores, and you focus on a subset, let's say the first 1% of the ranked database. You calculate the number of hits in the first 1% and divide it by the number of molecules in the 1%, and compare it with the baseline, so number of actives in the divided by the total number of molecules. So you can see how much enrichment, how many times can you identify actives in the top compared to the baseline. And this is used to validate the docking before running the virtual screening campaigns. And what I show you here is this enrichment factor. We did a molecular dynamic simulation of 5-HT6 receptors starting from a homology model, and we identified snapshots or frames from the MD run in a systematic method, that's the S, and with clustering, that's the C. And as you can see, the enrichment factor changes quite significantly. And in some cases, when we had the snapshots just next to each other, one of them could produce high enrichment factors. It was more than 15 times higher outranked actives. And then the next frame just couldn't produce any docking mode. And this is due to the fact of the movements of the protein. It was a whole system with a lipid bilayer, with water molecules, and with a docked ligand that was simulated with molecular dynamics. And we identified the rigid conformations of the protein, and then tested whether it can enrich in a docking the actives over the inactives. So the next level when you take the flexibility of the protein into consideration as well. There is a method called induced feed docking, when you dock the molecule and then with relaxed van der Waals radii, and then relax the protein structure in a consecutive stack, and then redock again. Or you can run MD simulations, have an ensemble, and calculate the binding, the ranking based on this ensemble. But free energy perturbation, simulating the whole system with an MD simulation is getting more and more attention. And although the theory itself is available for 60 years now, in the last decade or last 15 years, this evolved a lot. In case of free energy perturbations, there is a thermodynamic cycle. You simulate the ligand and the protein in solvation. Free energy perturbation will predict the delta G binding, so the energy change. You need a ligand where you know what's the binding affinity, and you are interested in a change of that ligand. How will that affect the binding affinity? You simulate the system when it's not interacting with the protein, both the starting molecule and the final molecule. And you do the very same with the protein ligand complex. And from the delta GB minus delta GA, you can calculate the delta G binding of the molecule. So how this is done? There is an alchemic transformation when from the real starting state, the system goes through series of non-physical states that's controlled by this lambda value, how much of the change is introduced to the system. The whole system is simulated with molecular dynamics. Let's think of a changing of a hydrogen to a chlorine, and you change step by step the van der Waals radius and the charge through dummy atoms from the starting state to the real final state. And then from this simulation, you can calculate the delta G. One method, this FEP+, that's from Schrödinger, that's applying OPLS 2 force field, uses a Hamiltonian replica exchange method in a special way. That's since here the sampling will be a problem, the protein binding pocket is heated up, the rest of the system stays cold, and they programmed it in a way that GPU processors can be used, and a system of 6K atoms can be simulated within 6 hours. So four steps can be done in a day. And here I'm focusing on the results. So FEP could produce on average, so they simulated 3,000 ligands, 3,000 endpoints predictions, and the average root mean square error was about, or the median was 1 kcal per mole. That's what they could achieve with this technique. So this means that if you simulate the system with FEP, with free energy perturbation, you know where to start, you know the initial state's binding energy, then a change can be accurately predicted if a high quality crystal structure co-crystallized with the initial ligands is available, if you have reasonable expectation of the conserved binding mode across the series, minimal tautomer ionization state changes, when the experimental binding data is originated from the same assay to have the high correlation, and when the X-ray structure is from the same protein construct. So yes, with knowing the approximations, molecular mechanics, MD simulation, for sampling the replica exchange, there are tools that within constraints can result in 1 kcal per mole. That's the desired accuracy for drug discovery. But there are cases when protein structures are not available, but data is available, and that goes to the data-driven approaches. And I cite here this elements of statistical learning, what I suggest as a starting point if you want to go into this direction. With this quotation, in God we trust, all others bring data. And then introducing this topic, we'll start with some data. At AstraZeneca, in the early years, early 2000 years, no computational pre-screening was required for solubility. And that was the result what chemists made in nearly half of the cases. The compounds had pure solubility. And solubility is before any biological activity measurement. So these compounds could not be measured in some of the cases. And then they applied simple solubility methods, and they asked the chemists to pre-screen with the solubility model before synthesizing the compounds, and then this dropped to 28%, and the number of good or highly soluble molecules increased in their system. But drug discovery data is expensive, and first we need to extract the information from the observed molecules. One of the techniques for that is matched molecular pairs. In case of matched molecular pairs, we are looking in the database for pairs of molecules that has the same conservative region, and only one change is observed. So one transformation from a cyanide to a chlorine will be defined for this molecule, and then we look through the whole database and check whether there are other pairs with the very same transformation. And on these different conservative regions, we sum up these rules and calculate the delta P solubility, for instance. So here, this region in one pair should be the very same, and the moiety should change the very same. And we have number of molecular pairs with different regions, but the same in the pair with only these changes. And this is a very effective tool. So you can see for different molecules, if you change the hydrogen to OH, how that will affect the solubility. In this case, green tells that it increased the solubility, it was neutral, or it had a detrimental effect on solubility. This is simply a data analysis tool. If you change a hydrogen to a phenyl ring on different pairs, on 1,300 pairs, then you will see that the changing a hydrogen to a phenyl will have a detrimental effect on the solubility. We also did a similar study on another target, and you can see that the delta, the absolute delta, and the standard deviation. So this will be a population, and there can be rules with high standard deviation. You cannot see any trend, but there can be rules with really significant high difference in between the series of pairs with low standard deviation. And why is that important? Since, as I said, drug discovery data is expensive, and with molecular match pair, there is another opportunity, is that you can exchange information without disclosing the structures themselves. And in this paper, they describe this method. At AstraZeneca, Roche, and Genentech, they identified molecular match pairs. And then MedChemica aggregated this data, since only the transformations and the corresponding statistics was shown or shared. And then a grand rule base could be compiled with 0.3 million rules. And then all the three parties who were involved in building up this database could use this new updated rule set. So benefit of this data analysis tool is that you can share data without leaking structures themselves. And since the time is running, I will skip some of the slides. So we are still looking for this formula, but in this case, we discussed physics-based methods, and now we will focus on descriptors and machine learning approaches. And here I will start again with how you validate your model and how you predict the error. When a machine learning model is trained, or a QSAM model is trained, you have a set of molecules that you split into a training set and a test set. After the model is built, you can also observe after the model building what was the accuracy. This split can be done in 3 different ways. You can randomly split the molecules to training and test set, time-based splits, or neighbor-based split. In time-based split, the new compounds will be in the test set, and in the neighbor split, compounds with less number of neighbors will be in the test set. And when it was checked, what's the R square, the correlation of the test sets, what's the accuracy of the test set, then if you do the splitting randomly, you will have the highest accuracy observed on the test set. While if you leave out neighbors or you use this neighbor method, you will have the lowest R square. If it is a time-based split or a prospective screening, then it will be in between these two. But what we are looking for is really the prospective screening. So we want to develop models that can be used prospectively. So this temporal split or time-based split is a technique that you have. If you have date field in your database, you can also build models accordingly. Within the training set, if you want to see what's the precision, there is a method called cross-validation. When you split it into chunks, you leave out one of the sets for validation and do the training on the rest. And you do it for all of these chunks. So every and each molecule will be one time in the validation set. That's called the cross-validation. It can be, it can have different faults, different pieces can be generated. Even one compound can be left out, leave one out validation, and then you can screen all of them. The other method is the bootstrapping, when you have these XY pairs and you produce with replacement or resampling smaller subgroups of your observations. And then you can calculate the variance, for example, of your model. So in this case, in bootstrapping, you have a whole collection and you generate one set that's a smaller set by picking randomly. And then you can pick again randomly. And then you can, with bootstrapping, you can have multiple infinite number of subsets. And on this subset, you can train the model and you can access the accuracy of the model. And there is a third method that's called conformal prediction, not to be mixed up with, it's nothing to do with 3D conformation. When you split the training set into a proper training set, you build a model on it, you calculate the residuals, the errors, and you build a model on the errors themselves. The reserved part goes to calibration set, and then you apply the model, you apply the error model, and then you rank the compounds based on the error, and look for a calibration factor that will ensure that's a simple multiplier. How much do you have to multiply the error to be able to predict in 80% of the cases the observed error on the calibration set? So you rank after on the calibration set the delta values, since you know the observed value, and then looking for a factor that at 80% or a given percent of the cases, the error multiplied with this alpha factor, the error model outcome, will cover 80% of the cases, and then you can have an error prediction as well. And still about the validation of the methods, classification is also, so in data, in prediction in general, there are two ways: regression and classification. In case of classification, in binary classification, the confusion matrix is used. You have actual positive and negative, predicted positive and negative. And you can calculate accuracy, what was the successful predictions compared to all of them, or balanced accuracy, sensitivity, that calculates the two positives divided by two positives plus false negatives. So from this confusion matrix, you can have different readouts that specifies the accuracy of your model. And here, what I recommend to use is the Matthews correlation coefficient, which takes into consideration all of the parts of the confusion matrix. Matthews correlation coefficients will be in between -1 and +1, anti-correlation and correlation, and 0 means no correlation. And that is to in real life cases, the number of positives and negatives in your sample is unbalanced. In most of the cases, like the positives are with lower population compared to the negatives. So a model that only provides you a negative might be perceived as a good accuracy, although none of the positives were found. And Matthews correlation coefficient helps you in judging models whether what's their accuracy. And there is another way. Paul Sodorowsky published a paper on this count on kappa, which is also taking the baseline into consideration. That's another method to balance. Since if we are building models, we have to know whether it just recognize the patterns in the training set, or what will be the prediction power of the model if it could deduce real relationships in the data. And there are pitfalls, and nice models can be generated for the training data itself that will have no use in real life. So we are still looking for the formula that will be a function of the descriptors, in this case, not physics-based rules, to predict the affinity of the molecule or any biological readout. The descriptors are representations of the molecules that is fed into QSA equations that can be physics-based, like lipophilicity, pKa, solubility, or topological polar surface area, different ring counts, bond counts. So these are the vectors that are fed into the system. And as ChemAxon, we are providing wide array of these type of descriptors. And another way to generate a descriptor set is the fingerprint technique. I don't know if you have ever heard about this. Fingerprints can be predefined substructures of the molecules that are coded at a given bit position. It can be pharmacophore-K based. So if there are hydrogen bond donors or acceptors, you can define topological distances and then code it into a fingerprint, or there are hashed fingerprints. In case of hashed fingerprints, there are two major types, the linear. Imagine a molecule like this. We define parties of the molecules from 0 to up to 5, 7 length. And these passes are deconvoluted from the original structure. And we use a hashing function that will map this string to a bit position. So we will have a 0-1 bit array of the molecule that is derived from the passes, the small passes available in the molecule. The first one will just code atom types, and then 1 bond length passes, and then 3 bond length passes. All of these are calculated for the molecule, and then based on the hash function, you can define the length of the bits themselves. Another technique will do the very same, but not with linear passes, but radial passes. So a nitrogen, a 1 bond pass, or a 2 bond pass will be decoded into a fingerprint. The difference in between the linear and the radial is that linear is substructure preserving. So if you have a molecule that's substructure of another, then all of the bits will be present in the target of the query, and that's not true for radial fingerprints. These fingerprints are quite effective for machine learning, since it can define chemical similarities, chemical moieties quite efficiently, and they can used in machine learning in multiple ways. Now, if we have this bit string, we can calculate similarity in between the two, like what's the shared bits that are common in both, and what's the total number of bits in the two bit strings, and that will be the Tanimoto metric. If the intersection is 19, we have in this case 24 in A and 21 in B, then you can calculate the similarity values. This is really an effective tool. There are various other ways how you can compare two fingerprints. You can define Euclidean distance, or a distance that takes into the consideration the shared bits plus the maximum of A or B. And this can be used differently, depending on the task you want to achieve. I'm just showing you some quick examples, what's the effect of this. So now, with a simple molecule, we are collecting similar compounds from a database that's 20 million compounds. And this is a histogram. This is in log scale. If you have Tanimoto metrics, this will be the different dissimilarity values. This is with Euclidean distance. And as you can see, the profile is quite different. You can also calculate the Manhattan distance. That's the difference count in the bits. Or what I introduced, if we have linear fingerprint or a radial fingerprint, it will have similar course of the curves, but at 0.6, in this case, we have 100K molecules, while at 0.6 with ECFP, we have less number of molecules, and there are some differences here and there. So you can calculate it for a single molecule, or you can overlay calculation in a cross screening of multiple molecules. What we did in this case, we compared GDB13 to a subset of drugs, and this is a colored histogram, a heat map histogram, where one point is colored according to the population at that point. So this is the cumulative distribution. This tells us how many molecules are at a given dissimilarity range to my query. And then we did it for multiple queries and then summed that up. So here you can see that, for instance, this point represents that at this dissimilarity, only 100 pairs could be identified, and that helps in judging the overlay or overlap of two compound collections, and tell us how many molecules can be selected with a given dissimilarity threshold. And you can also calculate this as a dissimilarity overlap in between, in this case, the vitamins. So you can use this dissimilarity value as a heat map as well. If you summarize the edges as providing higher similarity than a given range, then you can have a different view of your compound collection. In this case, this is a non-diverse set. Most of the molecules are higher than 0.7 Tanimoto similarity, and it's colored based on their property, the QAD score, how much they are fitting into the drug space. So it is non-diverse and not high-quality database. The other, we have less contacts, so less molecules. And each molecule in this theoretical example is a dot. we have less molecules that are connected to each other. So this is a diverse set with low quality. Here the color is more red, so it has higher quality, but still diverse. We have clusters, two main clusters and some outliers. And this is the desired with high quality and with high diversity. So this is the point. I think my presentation will be longer than expected. When we will turn, after we know the descriptors, the model building techniques. One of the techniques, there are different ways. You can apply linear regression, decision tree, support vector machine, or a neural network. In case of decision tree, to tell how it works as a basics, decision tree or classification and regression trees are working in a way that if you have a potential field like this, you want to build a model on it, and you will apply series of decisions. Imagine that you have samples on this surface. So the first decision is that the xy should be lower than t1, and then you can further make a decision that if x2 is lower than t2, then it will go into R1 bin, otherwise it will go to R2 bin. So based on the populations and the x1 and x2, the descriptors, you are splitting the data in a way that you want to maximize the classification accuracy. So if it is higher than T3, then you can divide by x2 at T4, so you can classify your sample. This is called decision tree. It's a really simple method, but its simplicity makes it also quite robust. It has two major hyperparameters: the depth of the tree and how many observations should be in one leaf, in one final leaf. Now, if you have a function like this, and if you do a card-based prediction, then it will define one. If you apply this random or this decision tree on your sample, then it will have three groups. But here comes this bootstrapping, what I showed you before. What if you sample your entire samples, subsample it, and you build different decision trees? Then you will get these plots here. So the different decision trees will result in different decisions where your actual predicted value is. Here the gray ones are left out from the prediction. And then from this example, you can have, from these weak learners at the end of the day, you can have an ensemble prediction. And with this ensemble prediction, you can even predict a continuous function. And as you have more and more of these weak predictors, of these weak decision trees, finally you can fit an equation, and the error will decrease until a certain point. And if you have a large number of cases, then it will not decrease any further. This is from the decision tree. This is called the random forest, when you have multiple hundreds of these decision trees that are built on bootstrapped subsamples of your original data set. All of these decision trees are providing predictions that are finally averaged, aggregated, and then you will have the final prediction. Here, one pitfall can be that if you have low number of observations on the final nodes, then the mean squared test error might be higher than if you have higher number of observations, since this will produce a shallower tree, so it will not go that deep in the decisions. It will not train for every and each observation individually. But if you have low number of observations, the tree will have highest depth. It will be more fitted to the problem. It will be overfitted. So on the training set, it will look perfect, but on the test set, it will have much higher error. That was the decision tree, and there is another algorithm called support vector machine that's used in pharma industry as well, where the aim is to fit a hyperplane that can segregate. Support vector machines was originally developed for classification problems that can classify the observations in a way that it applies a margin where misclassification is allowed. And that's a crucial benefit of it, that it can take some of the samples, if they are in the margin of the fitted plane, into account to achieve a better model quality. This works in a way that the algorithm places support vectors and then fits the hyperplane for the separation that's based on this is perpendicular to the support vectors in your system. And it can be also applied for 3D non-separable problems. As I said, we have large number of descriptors. You can imagine the fingerprints as being the descriptors, where it cannot be separated. And in this case, in case of support vector machine, it can map it to a higher dimensional space. And on the higher dimensional space with the support vectors, it can classify the observations. And the very same can be done in case of support vector regression, where we are fitting a plane. But in this case, a hyperplane, but in this case, the error margin should include the observations, and we can leave out some of the observations from the system, and that's the hyperparameter of support vector machines. And the third technique, and this is the last technique for today, what I want to introduce is the neural networks. That's AI and machine learning, and I have seen that there were also presentations about neural networks and how to teach neural networks. In case of neural networks, you feed your descriptors into the input nodes, and you have that many input nodes as the dimension of the input vector itself. You assign weights to these inputs, and then summarize these weights and use a bias term. And then this value is processed with an activation function. And then you will get an output. The activation function can be a logistic function, or there are different functions that will process based on this U value here. Now, these activation functions can be organized into multiple layers, and there can be a large number of hidden layers as well in the system. So you will get a matrix of the weight factors. And the teaching of neural networks works in a way that you have an input with a labeled data, with an observation. You have an output that starts with random weights, calculates the output itself, calculates the difference in between the observed and the output value, and back propagates the error throughout the neural networks in a way that you can have partial derivatives per weights. You can use, in this case, it's an optimization problem that's based in molecular mechanics and in various other areas. Steep descent can be used, or any optimization methods that will optimize the weight functions, the weights with this loss function, which is the delta in between the observed and predicted. So this is how this looks in a more complex scenario. You have the input layer, you have these weight matrices, the layers, and the output layer. And There are various hyperparameters, what's the backpropagation learning rate, how much of error should be taken into consideration in one step. And the optimization is done in most of the cases with backpropagation and in batches. So you submit the first batch of samples of observations to the system, and then you refine the weights, and then another batch of the observation, then again refine the weights, and you try to minimize the error through the weight factors. So now we have different techniques: neural nets, support vector machine, card trees, or ensemble trees, and they are having different characteristics. The prediction power is the highest for neural nets and SVMs, since there are more fitted parameters and these are more complex. But decision trees are preferred in pharma, since it can handle missing values. It's robust against outliers. It can handle mixed data types. For SVM, since this is in vector space, you have to normalize all of your inputs data. And there was a challenge, this TOX21 challenge, where a large number of structures were offered for prediction, and 650 compounds were added as a test set. And the group from Linz won this competition, and they applied deep learning, neural network, support vector machine, random forest, and also elastic nets. This is the prototypical cascade of virtual screening. It starts with a data cleaning and quality control, then chemical descriptors are generated. That's what I introduced, ECFP fingerprints, topological or even quantum mechanical. And then you select the model training strategy, evaluate the model, the area under the curve, and you can generate ensemble predictions as well. And in this context, it's worth to be mentioned that link this fingerprinting technique with machine learning. Fingerprints are like the pixels in an image, and in a neural network, in the different layers, a combination of these bits can produce high activation of a node that will correspond to a chemical moiety built up from different fingerprint bits. And then if you progress in these layers, that can potentially construct chemical moieties that are responsible for a toxicological outcome or for an activity prediction. And in this article, they describe that that's what happened indeed, since these molecules had the highest activation in the first layer, in the second layer, or some of the subsequent layers. And it turned out so this was a toxicological prediction challenge. And in the machine learning, based on the fingerprint technique, in the neural network layers, the activation functions for this, based on the activation functions, it can be constructed. What are the toxicophoric elements? And toxicologists who had knowledge from the past verified that these are prototypical toxicophoric elements. So the system learned from the fingerprints the knowledge of what the toxicology observed previously. And in this TOXCAX challenge, you can check the article here. The best method was the deep neural network, and then support vector machine and random forest if you consider the average precision of the models. So it was a long introduction, and yeah, I am a bit longer than expected. But this is not just used in competitions. This is from a publication from Bayer. What they are routinely using as a toolbox for absorption, distribution, metabolism, toxicity, and physical chemical property predictions. And you can see here the endpoints. I don't want to go into the details, but this is a nicely colored heat map. And how they are using it retrospectively. And recently, these are the techniques that they are using on these data sets to come up with predictions. As you see, support vector regression is used. Random forest is still the most popular or the most highly used, since it can tolerate outliers. But multitask neural networks also appeared and used more and more in pharma settings. So in pharma companies where there are a large number of data points available, machine learning techniques used routinely, and these predictions are channeled back to the design space so that the medicinal chemists can rely on these predictive properties. And that goes back to the Rubik cubes analogy. So this array of predictions helps the scientists to come up with balanced properties in their new designs. At ChemAxon, we are also developing a tool set for that, which is a training module where you can feed in your in-house data or even public data. The tool will build will do the descriptor generation, model building, validation automatically, and it can persist the model so that you can execute it later on. For feature engineering, we are using ChemAxon descriptors, but also user-defined descriptors can be introduced to the system. And we have selected random forest, since that's the most general purpose model. And although it cannot resolve these interactions, what I showed with the ToxCast challenge and how the toxicophores were deduced from the neural network. So in this case, all the dimensions are evaluated separately. But this is the most general method. So we are using random forest and support vector regression and on a pre-optimized hyperparameter set. And the tool can generate the validation statistics automatically. It can calculate the applicability domain for a new prediction. What are the most similar structures in the training set? And we are applying this conformal prediction to predict the error. And for overfitting, we selected ChEMBL-Y method. In case of ChEMBL-Y, that's again a simple trick. You have your database and the observed observations, and you randomly scramble the observations and try to build a model for that as well. Now, if the model is overfitted, then it will be able to fit on random values on the training set. So while you are training it, it will be able to reproduce the random collection to some extent. So for that, we introduced this ChEMBL-Y method. So you can check if the training itself has too many dimensions, too many descriptors, so that it can fit for a noise, a good model as well. And we used this model in an application study on 168 ChEMBL targets. ChEMBL is the bioactivity database publicly available, the largest. It has 1.8 million structures, 15 million data points, and we fetched 164 ChEMBL targets with from 500 to approximately 5,000 data points per target. Split it into 90% test training for the target of P activity, and we measured Pearson and root mean square error, and that was the results of our test. In more than 50% of the cases, higher than 0.8 Pearson correlation coefficient could be achieved. So with this training engine on publicly available data, predicting P activity only purely data-based, we could achieve 1.7 P activity root mean square error on this large diverse set of targets using random forest as a technique. And that's one of the examples, the HERG, which is a cardiotoxicological endpoint, and this is the test set. You can see the 0.5 and 1 bins error bars on this graph, and these are the statistical parameters. So in this case, in 72% of the cases, we could achieve P activity prediction in 0.5, relying on curated data and random forest techniques. And the last challenge, and I will wrap up quickly, is the data volume. So we discussed that the binding interactions are complex, the human body is complex, data mining can have its own pitfalls due to the overfitting. But there is another challenge, what to predict. What are the molecules that you want to screen? And in this field, there was a large expansion. The collections that can be screened and that vendors are offering for screening for pharmaceutical companies increased in the last 5-8 years quite significantly. Now, Enamine offers a real database that has now 1.2 billion structures, and they claim that they can synthesize these compounds. These compounds are not synthesized, but they have the receipts, the ways to synthesize if one of them is needed. So it was virtually enumerated, and they claim 80% of shipping these compounds. So the pharma companies now can select from 1 billion compounds. And yeah, quantum mechanics are to be applied for smaller systems, looking for up to millions of structures. But if you have billions of structures, that places the whole challenge into a different dimension. And for that, we again have to step back and apply different methods that are not even predicting the properties, but just telling us if there is a similar compound in this large data set. And fast similarity searching done in memory is one of the tools that can cope with this. So in the ChemAxon MedFast similarity search tool, you have a structure file, you generate the fingerprints, and then load it into a server so that it will be a full in-memory search on fingerprints. And these are just some of our results. With similarity searching, we rented an Amazon cloud EC2 instance that has 1 terabyte RAM, loaded the 1.2 billion structures using chemical fingerprints, in this case 500 bits long. Pre-processing took 6 hours, and the server can be started up in 7 minutes. And here you can see the average search type on 1.2 billion compounds. If you want to retrieve the top 100, it can be done within 2 seconds, in 1.16 seconds, measured with 500 runs. This average. But if you want to collect the top 15K most similar structures, for a query molecule, that's also less than 2 seconds. So in less than 2 seconds, from 1.2 billion structures, you can collect the most similar. The other way is to surf in this large set is the substructure searching. When you want to collect the molecules that are having a chemical moiety. Substructure searching also applies a pre-screening with linear hashed fingerprints, fingerprint match, and also descriptors like the molecular weight or different counts are equivalent. And then this is a graph, an NP-hard problem, a graph isomorph check. The subgraph of S, a graph S is subgraph of a graph G if S is isomorphic to the subgraph of G. So it's the substructure relationship, and there are different methods, the Ullmann method, VF2 plus, how to do the mapping of the graph elements. And also, we tested our JChem PostgreSQL cartridge, which is a full-blown database cartridge. We loaded the 1.2 billion structures, again, on a commercially available AWS instance with 20 worker loads, 16 nodes for each, and measured the search types, the search speed, and on this set, a full structure search could be done within a second. So this is milliseconds. A similarity search was way below one second. And the very last point is how this is utilized in drug design. In drug design, you have a large number of services, match molecular pairs, QSAR, docking, AI, machine learning, and the designers should be able to access these services. So you have to connect these bits and pieces into a design space. And our tool is the design hub that can utilize different services, compound catalogs, Fischem predictions, custom activity predictions, and connect it with the design. So whenever you are making a change in your hypothetical compound, then it can calculate with random forest, the hERG, the calculated properties from this enamine real, the number of analogues that are available, MMP, or even docking results. So the last challenge is how you project all of the models on the desktop of the medicinal chemist so that it will be utilized in design. So that's the last slide, last but one. So in summary, sorry, I know that the presentation was quite fast, but I tried to have simple analogies in it. There are challenges of the complexity of the human body, the binding interactions, the pitfalls when we are dealing with data and the large space of the chemicals themselves. And the last is how all of these bits and pieces of the models are projected and connected for the designers and medicinal chemists so that they can consume these services, they can utilize the predictions. So there are a lot of challenges and opportunities on this field, and I hope that you could follow my thoughts mainly. And the last slide is essentially all models are wrong, but some are useful. Pharma companies are looking for these useful models. Thank you for inviting me, and thank you for participating in this talk.